What's happening, guys? Aruj again, and today we're talking about gene splicing and recombinant DNA. Before we get into the process, let's go over some basic vocabulary. So the first one that we have is genetic engineering, sometimes called genetic modification. And all that means is to change the DNA of an organism purposefully, to do it with intent. Essentially, it's splicing genes from one organism, inserting it to another one, and when we do that, that organism that has new DNA, we call that a transgenic organism, sometimes called genetically modified organism, or GMO for short. So think of the example of the cat that glows in the dark that we talked about in class, the glow-in-the-dark cat. Well, that cat has a protein from a jellyfish, or I'm sorry, has a gene to make the protein from a jellyfish. So that cat would be called either transgenic or a GMO. When we talk about recombinant DNA is the DNA that transgenic organism or the GMO has. That DNA has been recombined. Okay, that DNA has been artificially made by somebody in the lab, where they take pieces from different organisms and recombine them together. So the DNA that they have is called recombinant. Okay. Now, how do we actually cut the genes out? Well, we use what's called restriction enzymes. Restriction enzymes will cut segments of DNA at very precise locations, at very specific areas. EcoR1 is one type of restriction enzyme, and you'll see that on the next slide over. And when restriction enzymes are used, they always have what's called sticky ends. And what the sticky ends are is at the two ends that's cut, there's nucleotide overhangs, meaning there's nucleotides that aren't paired. They don't have their complementary bases. So they want to attach to another base. You'll see why that's important in just a second. Genetic engineering is very common to use bacteria because bacteria grow very, very fast and it's easy to replicate those genes. Also, they have circular rings of DNA that are called plasmids that are pretty easy to work with. And you'll see what that looks like in just a moment. Scientists are able to use plasmids and reinsert that into bacteria to make large amounts of human proteins. So insulin, growth factors, growth hormone, blood clotting factors. And why is this important? Why do we use this? Well, a lot of these human proteins, like insulin and growth hormone, those are things that are prescribed by a doctor. So pharmaceutical companies can make them this way. It's very fast and very cost effective. Before, in the past, if somebody had diabetes, they would have to get insulin from other animals like pigs, and now we could grow identical insulin that's the same as human insulin from bacteria, and it's the same that our body already produces. Okay, so let's look at an example of how they do this with growth hormone. So first we take our human cell and a bacteria cell. And this right here, this circular ring, that's the plasmid. That's the piece of DNA that the scientists are going to modify, okay? But before they do that, they have to find the gene. They have to find the segment in the human DNA that codes for growth hormone. So they isolate that first. They find it. And then they give it the restriction enzyme. In this case, it's EcoR1. They do the same to the plasmid DNA, or the bacterial DNA. They expose that to the restriction enzyme. Now watch what happens when, remember this is the restriction enzyme, watch what happens when it cuts the DNA out. It leaves the sticky ends. It leaves these unpaired nucleotide bases, okay? And what's unique is they're complementary of each other. So they want to pair with each other. The growth hormone, the gene for growth hormone, wants to pair to the bacterial plasmid. And so they do. Now, this circular ring of bacterial DNA is now called recombinant because it now has that gene for the human growth hormone. And now what the scientist does is they insert that back into the bacteria, they put the plasmid back in, and when this bacteria replicates asexually, okay, when it splits into two other bacteria cells, each offspring is going to have that gene for human growth hormone, okay? Yeah, so it's a short one for today. I do want you to make sure you get this slide in your notes as well as the vocab on the previous slide. And that was it. See you guys next time. Bye.